Okay, we're going to get started with Pillar 1. So remember, Pillar 1 is replacing maternal UBE3A. So our first speaker in Pillar 1 is Dr. Barbara Bailus. She's coming to us from the Keck Graduate Institute, and she's going to be talking to us about the Backstage pass to the brain for therapeutic delivery. Barb is the chair of our scientific advisory board, and I just want to let you all know that Barb has become one of the most impressive volunteers that FAST has ever had. She puts time, effort, money. I wake up every morning to text messages about new discoveries in therapeutics unrelated to Angelman syndrome, and then she wonders within about 15 to 20 minutes why I haven't responded to her text. And it's because I have 500 emails I also need to respond to, Barb. But she is always thinking about Angelman syndrome. She doesn't live with Angelman syndrome. She kind of lives with me. Therefore, she lives with Angelman syndrome by association. But she is one of our biggest advocates, and she is a non-parent who cares deeply. So, Barb, thank you.
Okay. Thank you for the kind intro, Allison. I'll give you 20 minutes next time to respond to those emails. All right, so today I'm going to talk to you mostly about delivery aspects. So that's why we have Backstage passes to the brain for therapeutic delivery. A little bit about me. The technology that you're going to hear today, I invented back in my postdoc, so that's what you do after you get your PhD, with Dr. Lisa Ellerby. I have since brought it forward to apply to Angelman syndrome. I'm an assistant professor at Keck Graduate Institute. That's the Claremont Colleges in sunny Southern California. And I'm dedicated toward finding a cure for Angelman syndrome. So I started my journey in Dave Siegel's lab at UC Davis, working on zinc fingers. And then I moved on to the Ellerby lab, where I worked with CRISPR and how to deliver CRISPR. And then I eventually wound my way to the Keck Graduate Institute, at which point I had my own lab, and I decided to study Angelman syndrome because of the community and because of how amazing it is. So I think we've seen the pillars here. We've got pillar 1, 2, and 3, and 4. And I'm going to focus today on pillar 1, replacing mom's gene. Now, we can do this by giving back not just the DNA, but actually skipping over the DNA and giving the product of the DNA, the protein. And the way that we can think of this is called enzyme replacement therapy. The UBE3A protein is an enzyme, so that's why we call it enzyme replacement therapy. And you can give pure UBE3A to the brain. You would do this by infusing the UBE3A directly into the brain. It's a little bit invasive. That's probably an understatement. So the idea would be, can we do this better? Instead of doing direct infusion to the brain, can we instead give it as a vein injection, just like an insulin injection or an insulin pump? And so this is where the delivery system that I'm going to talk to you about comes in. I call it zip delivery. The reason we call it zip delivery is because we took a teeny, tiny piece off the Zika virus, which is known to infect neuronal cells, hence brain cells, and we attached it onto a therapeutic protein. And so I think of this as a backstage pass to the brain. The cool part is I can give this to other proteins besides UBE3A, but for today, we're going to focus on UBE3A. When we first did this, I was working in Huntington's disease in the Ellerby lab. And so we put it onto a CRISPR nickase. And this is really great for Huntington's disease. It's not great for Angelman syndrome, so I'm going to tell you how we had to change. But I want to show you some of why this was so amazing. So we put this into the tail vein of a mouse. That's sort of like your arm vein here. And it got all over the brain from a single injection. In an adult mouse, these mice were 16 weeks old. That's kind of like a 16 to 20-year-old individual. So in our untreated mouse, you don't see very much red. In the zip CRISPR mouse, you see a lot of red. That red signal was the CRISPR. And when we zoomed in on the hippocampus, you can see a lot of red. This was still focusing on Huntington's disease. So when I started my own lab, I came back to Angelman syndrome, and I thought, what if we could put that backstage pass on UBE3A? And so that's what today's work is going to focus on, adding that backstage pass to the UBE3A. Why do we need to do that? Well, as Ed mentioned, we have something up there called a blood-brain barrier. I think of it as the big bad bouncer. It does a really good job for most things, but every once in a while you want to sneak something past him. Unfortunately, UBE3A on its own, not so good. But if we can slip that backstage pass onto UBE3A, we could get past the blood-brain barrier. And so I've started doing this work in my lab for ZIP UBE3A. We've put the backstage pass on, and then we've also put on something called a flag tag. This is important so that you understand how I'm able to visualize this UBE3A. So this flag tag relates over here to this black bar. You can think of that as like the selfie for UBE3A. When we see this show up, it is there. Now, this protein is actually produced by a company called GenScript. And this is great because it's one step forward to consistency and reproducibility. They've done several batches of this now for me. We've taken this UBE3A and we put it on neuro2A cells. These are mouse neuronal cells. It's important because I'm going to have to go into the mouse very soon. So I need to know that this gets into mouse cells. Now, what you see here are a bunch of black smudgy marks. But those black smudgy marks are actually the UBE3A with the ZIP backstage pass and the flag tag. And you can see at 4 hours and 24 hours it's in the cells. The NT means no treatment. What's also really important here is you're seeing the UBE3A get broken down. Now that sounds a little odd at first, but we want to see that. We don't want our new UBE3A to just accumulate in the cell in an unlimited amount. So the fact that it's naturally breaking down is very important. We can also see a slight shift in size. So these cells already have UBE3A. So in that second middle panel, you see a bar that's just above the really dark ones. That is the artificial UBE3A that I put in. It's a little bit bigger, so we can see the difference. We repeated this in human neuronal cells because as great as it is to cure mouse cells, I really actually do want to cure the humans. So when we do this, you can see exactly the same result at 4 hours and 24 hours. We have the protein in there. And we see that same slightly larger UBE3A. So this is exciting, and this is going to take me forward to mouse studies, which is going to happen in the next 6 months. The next question that I asked, and I did this in collaboration with Dr. Albert Kung, is the UBE3A that we've made that can get into the brain actually working and doing its job? And so this figure's a little complicated, but you have what's called ab nova UBE3A. That's UBE3A that I can purchase commercially. And then we have our UBE3A with the flag tag and the backstage pass. What I want you to focus on here are the 2 bars that say 100. Those are the tall bars. It's measuring how active the UBE3A is in doing its job. And what's really nice is that you're actually able to see that this new UBE3A with the ZIP backstage pass performs as well as what is commercially available. So this backstage pass does not inhibit the UBE3A activity in any way. We're also testing these in organoids, so this data should be out in another month, but they have actually added this protein in Dr. Albert Kung's lab onto the Angelman organoids. So those are those mini brains, because I can't use brains in a dish, so we do the next best thing and have mini brains when we're adding it. And as mentioned, they've actually already made the next batch of protein for mice, so we're going to be testing this probably in January or February, and I'm hoping that by next year when I stand up here in front of all of you, this is not a cartoon image, but that this will become real data, and I'll be showing you this with widespread UBE3A delivery. And then we are looking at something beyond ZIP, so I'm categorizing new backstage passes because we might be able to find something even better than ZIP. And the reason we're doing this is, well, we should have all shots on goal. This should be occurring in mice in 2025 as well. And then I'd like to take a moment to thank everyone who's made this possible. So the Bailus lab, pretty much you wouldn't see anything up here if I didn't have this fantastic lab. The Ellerby lab, where I invented the zip technology. The Kung lab, who did an amazing job with the activity assay. And then a special thank you to Dave Siegel, where my Angelman syndrome journey began. A special thank you to Allison, who got me back into the Angelman field after my postdoc and has been so incredibly supportive. But most importantly, a thank you to all of you, the parents in the audience. You funded this study. You have continued to fund this study, and it is all of your hard work, your lemonade stands, your golf tournaments. That is what helps actually make these studies possible. So you actually owe yourselves a round of applause for making this work possible. Thank you so much.
Coming over, one second.
What's that?
The other people in the audience will want to hear.
Thank you, Dr. Bailus, for the work that you do for our kids, first of all. And this might be a little bit too early, but given the administration, is there any thoughts on how transient this would be, given it's like an enzyme replacement, that there would potentially need to be multiple administrations? And then, if that being the case, is there data on the Zika virus if there's an immune response with that subsequent administration?
Yeah, so these are all really good questions, and so we're going to be answering a lot of this when we go into the mice. For the immune response, I don't have the answers to that yet, but we are using a very small piece of the virus. And for repeat administrations, this would be something where you would have to do repeat administrations, but the positive part here is that those administrations would be IV injections instead of direct brain injections.
Okay, great, thank you.
Dr. Bailus, you're amazing. Is
Thank
this
you.
something, given Pillar 1, would you envision this being done in conjunction with some of the other strategies? Is it an on top of, potentially?
Yeah, so I could definitely see layering this with potentially other strategies or other therapies as well. So I think being able to combine is really nice, yes.
Coming.
Hi, just echo everyone's thanks as well, Bob. You said that we want the UBE3A protein to break down in the brain. Is that because too much would cause the build-up, such as the problems that we see with DUP15?
Yeah, so I think we wouldn't want to see the UBE3A just permanently accumulate. You could end up with something maybe like DUP15, although you would really, really have to have a lot of it. But it's also just that you want to make sure that it can be broken down, because like all proteins, eventually you need the protein to cycle through and get broken down, because proteins themselves eventually wear out. So we do want to see that any worn out protein would eventually get recycled.